Viruses we are born with. Human endogenous retroviruses, HERVs, are genetic elements derived from ancient retroviral infections that have become permanently integrated into the human genome over millions of years of evolution. These retroviruses, once capable of replicating and spreading between hosts, have lost their ability to produce infectious viral particles and are now considered an integral part of our genetic code. Despite being remnants of past infections, HERVs have garnered significant interest due to their potential roles in various biological processes and their implications for human health and evolution. Retroviruses are a unique class of viruses that possess the ability to reverse transcribe their RNA genome into DNA, which can then be integrated into the host cell's genome. When a retrovirus infects a germ cell, egg or sperm, or a cell that gives rise to the germ line, the integrated viral DNA can be passed on to subsequent generations. Over time, these endogenized retroviral sequences have accumulated mutations and have become fixed within the host genome, giving rise to HERVs. HERVs are estimated to account for approximately 8% of the human genome, with over 98,000 distinct HERV loci identified to date. These elements are classified into different families based on their sequence similarities and structural characteristics, with the three major families being HERV-K, the most recently acquired, HERV-H, and HERV-W. Each family is further subdivided into subgroups and individual proviruses. Initially thought to be mere junk DNA, HERVs have been found to play crucial roles in various biological processes. Placental development. Certain HERV proteins, such as syncytins, are involved in the formation and maintenance of the placenta, facilitating cell fusion and nutrient exchange between the mother and fetus. Gene regulation. HERV regulatory sequences, such as promoters and enhancers, can influence the expression of nearby host genes, contributing to gene regulation and potential phenotypic diversity. Immune function. Some HERV proteins have been implicated in modulating immune responses, either by triggering immune activation or suppression, potentially contributing to autoimmune diseases or cancer development. Neurodevelopment and neurological disorders. HERVs have been associated with various neurological conditions, including multiple sclerosis, schizophrenia, and certain neurodegenerative diseases, although the underlying mechanisms are not fully understood. Evolutionary implications. HERVs provide valuable insights into the evolutionary history of humans and their interaction with ancient retroviruses. By analyzing the distribution and diversity of HERVs across different populations and species, researchers can gain insights into evolutionary relationships, migration patterns, and speciation events. Additionally, HERVs may have contributed to human evolution by introducing novel genetic material and influencing gene regulation, potentially driving phenotypic changes and adaptations. Human endogenous retroviruses, HERVs, have been implicated in the development and pathogenesis of several psychiatric disorders, particularly schizophrenia and bipolar disorder. Although the exact mechanisms are not fully understood, there is growing evidence suggesting a potential link between HERV activity and certain psychiatric conditions. Schizophrenia. Several studies have reported increased expression levels of specific HERV families, particularly HERV-W and HERV-K, in individuals with schizophrenia compared to healthy controls. These elevated HERV expressions have been observed in various tissues, including the brain, cerebrospinal fluid, and peripheral blood mononuclear cells. Possible mechanisms. A neurodevelopmental impact. HERV proteins, such as syncytins, have been shown to play roles in neurodevelopmental processes like neurite outgrowth, synaptic formation, and neuronal migration. Dysregulation of these processes due to aberrant HERV expression may contribute to the neurodevelopmental aspects of schizophrenia. Immune dysregulation. HERVs have been linked to immune activation and inflammation, which have been implicated in the pathophysiology of schizophrenia. HERV proteins may trigger immune responses, leading to neuroinflammation and altered brain function. Epigenetic regulation. HERVs contain regulatory sequences that can influence the expression of nearby host genes. Abnormal HERV activity may disrupt the epigenetic regulation of genes involved in brain development and function, potentially contributing to the onset of schizophrenia. Bipolar disorder. Several studies have reported increased HERV transcriptional activity and protein expression in individuals with bipolar disorder, particularly during manic episodes. The HERV families most commonly associated with bipolar disorder include HERV-K, HERV-W, and HERV-H. HERVs may influence the expression or function of genes involved in mood regulation, neurotransmitter systems, and circadian rhythms, contributing to the mood dysregulation observed in bipolar disorder. Immune activation. Similar to schizophrenia, HERV-mediated immune activation and neuroinflammation may play a role in the pathogenesis of bipolar disorder. Epigenetic modifications. Aberrant HERV activity may lead to epigenetic changes that dysregulate genes involved in mood regulation, neurodevelopment, and neuroplasticity, potentially contributing to the onset or progression of bipolar disorder. It is important to note that the association between HERVs and psychiatric disorders is an active area of research, and the causal relationships and underlying mechanisms are still being investigated. HERVs may interact with various genetic and environmental factors, complicating the understanding of their precise roles in psychiatric conditions. Nonetheless, the study of HERVs in psychiatric disorders holds promise for elucidating pathogenic mechanisms, identifying potential biomarkers, and exploring novel therapeutic targets. The potential link between human endogenous retroviruses, HERVs, and depression has also been explored, although the evidence is more limited compared to schizophrenia and bipolar disorder. Several studies have reported altered HERV expression patterns in individuals with major depressive disorder, MDD, compared to healthy controls. Here are some key findings. 1. HERV-W and HERV-K expression. Increased expression of HERV-W ENV and GAG transcripts has been observed in the brain tissue and peripheral blood mononuclear cells, PBMCs, of individuals with MDD. Elevated levels of HERV-K antibodies have been found in the serum of depressed patients, suggesting an immune response to HERV-K antigens. Potential mechanisms. Neuroinflammation. Like other psychiatric disorders, HERVs may contribute to the development of depression by inducing neuroinflammatory processes. HERV proteins can stimulate the production of pro-inflammatory cytokines, which have been implicated in the pathophysiology of depression. Epigenetic regulation. HERV regulatory sequences can influence the expression of nearby host genes involved in mood regulation, stress response, and neuronal plasticity. Dysregulation of these genes through epigenetic mechanisms may contribute to the development of depressive symptoms. Neurodevelopmental impact. Some researchers have proposed that HERV-mediated disruptions in neurodevelopmental processes, such as neurogenesis and synaptogenesis, may increase susceptibility to depression. Immune dysregulation. HERVs may contribute to the immune dysregulation observed in depression, potentially through molecular mimicry or by triggering autoimmune responses. It is important to note that the association between HERVs and depression is an emerging area of research, and the findings are still preliminary. The relationship may be complex, with HERVs potentially interacting with other genetic, environmental, and immunological factors in the development of depressive disorders. Additionally, it is worth mentioning that some studies have failed to find significant differences in HERV expression between depressed individuals and healthy controls, highlighting the need for further research to clarify the role of HERVs in depression and replicate findings across different populations. Overall, while the link between HERVs and depression is not as well established as in schizophrenia and bipolar disorder, ongoing research in this area may provide insights into potential pathogenic mechanisms and contribute to a better understanding of the etiology and pathophysiology of depressive disorders. therapeutic potential and challenges. While HERVs have been implicated in various diseases, they also hold potential for therapeutic applications. For instance, HERV-derived proteins or sequences could be exploited as targets for novel diagnostic tools or therapeutic interventions. However, the complex interplay between HERVs and host genes presents challenges in understanding their precise roles and developing targeted therapies.